title of my research is Effect of Probiotic Dosage on the fish growth, water quality, and microbial composition in aquaponics system. And thank you for your patience and allow me to present this presentation. And the presentation outline includes introduction part, objective, material and method, and result and discussion and conclusion and recommendations are included in general outline. The background of this study is aquaponics is a sustainable food production system that integrates aquaculture and hydroponics. The aquaculture, which is due to fish farming, and hydroponics, soilless plant cultivation in closed loops recirculating environment. It implies the circular economy principles by minimizing waste and reducing input needs. Aquaponics can significantly improve fish food security, but its adoption in developing regions like Ethiopia remains limited due to high startup costs and lack of localized adaptation strategies. The system components and the microbial role includes African catfish. Different types of fish can be included, and the most selected here in Africa, especially in Ethiopia, is African catfish. Claris garipinus is widely farmed for its fast growth and nutritional value. Through its vulnerable, however, it is vulnerable to pathogens. In comparison to plants for hydroponics, lettuce is one of the most important vegetable globally. Significant seed crops benefits from fish-derived ammonia, making it ideal for aquaponics due to its short growth cycle and market demand. And the aquaculture sector, faced economic loss from the excessive bacterial infection with limited African research on fish immunity. Antibiotic alternatives are needed to protect fish and human health while maintaining sustainable. So, role of probiotics. The role of probiotics: enhance feed efficiency, improve gut health, stimulate vitamin production, and maintain microbial balance, thereby promoting fish growth and water quality activities. Such kind of benefits are observed under the probiotic study. So research gaps that observed identified in this study, and the objectives included such objective: establishing an aquaponic system using local material due to its needs high economic impact. In this study, designed to establish a aquaponic system using local material, isolates and apply gastrointestinal probiotic in various dosage, isolating from the same species and applying that isolated probiotic in different dosage. Assessment of its effect on the growth and of the fish and the lettuce, analyzing the water quality, nitrogenous compound cycles in microbial composition are assessed in this study. Major activities are set up the aquaponic system from local material. Promoting the probiotic screening is one of the major activities. Impact assessment is the other major activities. Monitoring this impact, this assessment are the activities. The material and methods, the general material and methods: experimental design, collect African catfish, isolate the guts, establish the aquaponic system, formulated feed, inoculate the probiotic bacteria. The probiotic bacteria that isolated, inoculated to feed at different dosage, and the stocked fingerlings that collected from the reservoir and stocked at the same stocking density, transplant the lettuce from the lettuce grow bed, and assessment effect on the fish and lettuce growth, water quality, nitrogen compound. The objective assessments are included under the methodology part. These are the gut probiotic analysis different from sample collection up to using the microbiological manual and Da Silva studies are the basic procedures that I used to isolate the microbial, the probiotic species. Purification of the probiotics, it has its own selective media to grow only the probiotic species like MRS agar and MRS bros are the most important nutrient to grow microbial, the probiotic species. And colonies are counted and purified, so the microbiological activity. Then the purification takes place by repeated activities, by repeatedly doing the subculturing. Screening, also the other activity, screening those isolated microbial, the probiotics. Morphological identification is there, biochemical tests like catalyst, SIM test, Gram-SAN, and other activities come included here in this study. Tolerance test also done, acid tolerance between the range of 2 to 4 pH, temperature tolerance also studied between 15 to 45 degrees centigrade. It is depend on the species. Bile salt test also done between 0.3% up to 0.5% of bile salt tolerance studied. Salt tolerance also from 2.5 up to 10% of salt tolerance are included here. And the key function that used to isolate these probiotic species for aquaculture, one is catalyst test, the other acid tolerance, because the stomach is acidic environment. Unless the species that isolated must be, unless they survive in the high acidic range, they cannot perform their work. Salt tolerance at the same time. Antimicrobial activity also, antimicrobial test. Inhibition zone is measured. Those higher inhibition zone recorded probiotics were selected. Cell surface hydrophobicity, also the most important, because inside of the gut, the attachment of the probiotic bacteria to the guts or the wall of the intestine of the fish must be identified. Those have high surface hydrophobicity, cell surface hydrophobicity are the most important to adhesion and for the immune interaction, since once the antimicrobial activity is done in their inhibition zone, so cell surface hydrophobicity is important. This the other points under the methodology, aquaponics system setup, since it is startup is need high cost. I used fish tank from wooden fish tank that's layered by plastic layer to hold the water, and it is fixed with water pump to pump water from fish tank to hydroponics to grow bed for plants or lettuce. Aerator also fixed to the each fish tank, because oxygen depletion may be affect one of the factors. So to reduce that, aerator is attached to each fish tank, and heater temperature regulation also one of the other factor. The constant temperature were controlled using aqua heater or temperature regulator fixed to each fish tank for stable condition. And solid removal tank is one of the tanks that required in hydroponics, one hydroponic system, biofilter, in which the ammonia converted to nitrates to the plant. Hydroponics units, plant grow area is also a unique part for hydroponics is locally prepared material. That is the plastic bottles collected from surrounding inside of the town that may cause environmental pollution that were reused for hydroponics grow bed for lettuce growth. In the circulatory system, the water can reach there. So the aquaponics system is setup of aquaponics system is this. And the other feed formulation, I used feed from 4 ingredients locally available and even as a waste product from low environment, like the poultry manure, cattle blood, rumen liquor, azola pinnata, and formulation also the modern using using a linear programming solver. This software used to formulate this feed. And total feed required is standard. 5% of their body of the fish is given for 60 days. Processing steps and premixing of the feed, each ingredient after identifying by linear programming solver. Premixing, mashing with water, pelletizing, sun drying, measuring, and packing are steps for processing the food. And the next is administration. Turbidity of the probiotics were measured, and using the most standard material is known as measurement, the Palin test photometer 7500 Bluetooth is the most modern that used to measure the turbidity. Turbidity measuring means counting indirectly, determining the number of the cells that found per ml. So in a study designed 25 to 2,500 cells in 8 ml is found for the first treatment. Second, 1:3 ratio were applied. The volume of the isolates. The final volume is also determined for treatment 1, treatment 2, treatment 3, and inoculated to the feed and supplied to the fish. Stocking the fish is the other 36 African catfish fingerlings were stocked at 6 stocking density. Water quality initially was measured. Feed protocol as 5% of their body weight. Feed adjustment were done at recalculated by 2 weeks as a standard. And lettuce transplanting to hydroponics, 192 lettuce seedling was transplanted to the hydroponics, and initial growth parameters like leaf height, leaf width, leaf number, total height, and total weight were measured. No significant difference initially. Lettuce growth was measured weekly for 5 weeks. And at the end of the experiment, survival rate of the lettuce, nitrate absorption, and overall growth of the lettuce was evaluated. And the water quality, nitrogen compound, microbial composition also from the water, and the tissue of microbes. The tissue, three tissues, gills, stomach, and skin of the fish, microbial load were studied. And these are data collection methods: the probiotic screening, growth conversion metrics, fish biomass production, water quality monitoring, nitrogen compound analysis, hydroponics lettuce, microbial composition. These are the data collection methods. The collected data were analyzed using SPSS ANOVA, SPSS version 20, and Duncan test were applied for comparison of mean comparison. And the result and discussion. For the first objective, dominant isolates were lactobacillus and lactococcus, and leuconos also. The 3 dominant species were isolated, of which 87.5 were gram-positive, and acid tolerance, acid and bile tolerance. All lactobacillus are tolerated the acid. One leuconos also survived as a pH 2 and 4, 2 to 4, and 0.3 bile. Only 12% of lactococcus tolerates a low pH. That means strong pH tolerated. So this similar probiotic profile reported in aquaculture by Melo et al. 2020 and Brown et al. 2021. The tolerance, pH, and bile also as gastrointestinal survival reported. Antimicrobial activity and adhesion potential. Adhesion or the attachment to the wall of small intestine of the fish. All isolates inhibited 6 pathogens, fish pathogens especially. Those are inhibited zone is ranging between 10 to 60 millimeter, which is compared with the standard for bacteria and fungus also. High cell surface hydrophobic lactobacillus contain 60% of cell surface hydrophobicity. That means according to the standard, the cell surface hydrophobic 0 to 29 is a low, 30 to 60 is a medium, and 60 above 60 up to 100 is high surface cell surface hydrophobicity. So based on this standard, my probiotics were found in the medium parts, in the medium arrangement. So antagonistic effects attributed to organic acid, bacteriocin production or studied by different acid peoples, but the cell surface hydrophobicity is not focused here. One of the other, the second objective. The objective to growth and feed conversion of the catfish. The higher growth performance were in T2, recorded in T2, followed by T3 and least in T1. Key indicators are here: weights gain, food conversion ratio, food efficiency, protein efficiency ratio, protein productive value, and specific growth rates also calculated. This under enhanced growth due to the probiotic mediated gut health and nutrient absorption, according to the comparison studies. And the third objective, influence of water quality parameters. Water quality in aquaponics system, dissolved oxygen temperature and pH showed no significant difference in all treatments, but electric conductivity, total dissolved solid, and salinity showed significant difference, variation, increasing with probiotic dosage. When probiotic dose increase, their value is increased. Under this comparison, improved nutrient mineralization may be increased when electric conductivity or TDS by Cruz et al. Stable pH and DO confirmation biological stability. This also stability of the DO and pH can be comparable. And the fourth, nitrogen compound management, nitrogenous compound concentration results. Ammonia, ammonium, nitrogen are higher in fish tank, and nitrate is higher in biofilter, because biofilter, inside of the biofilter, nitrate production is there, because there is substrates that are found in the nitrate that may be benefits for attachment of bacteria. And nitrate utilized efficiency higher in T3. In T3, nitrate utilization. Probiotic, and for the discussion part, probiotic enhanced nitrification, lowering toxic ammonia and increasing plant availability nitrate. Efficient nitrate uptake by lettuce supporting integrated system performance. And the objective 5 indicates lettuce biomass enhanced, lettuce grows with hydroponics unit. Result indicates T3 has the highest biomass, 672 grams, and the net production is 7 kilograms, almost 7 kilograms. Fresh leaf regrow post transplant in all treatment. Discussion part, better growth linked to nitrate uptake, supported by nitrifying bacteria and reduce NH toxicity, as NICA indicates. Objective 6, microbial composition of water. The result indicates fish tank microbial load the highest in T1, microbial load highest in T1, least in T3. Gut microbes higher in the gut, microbial load is higher in the gut than skin, then the least is a gill count lower in T3. When the probiotic dosage increase, the number of microbes is reduced. And for the discussion part, the comparison part, probiotic compete pathogenic microbes and suppress harmful colonization. Improved water and tissue hygiene in T2 and T3 treatment as indicated, based on the number of the microbial that found in the water and tissue of bacteria. In conclusion, the mixed probiotic, lactobacillus, lactococcus, and leuconostoc improved fish growth and feed efficiency, water quality, and nitrogen cycling. Lettuce biomass in hydroponics, reduce microbial load in system. For the recommendation part, T2 probiotic dosage yield optimal performance for integrated aquaponics system. This is one of my experimental place, and the result is, since the paper is on the under review, the result, this is the place where the system is studied. Thank you very much.